এইবার আওয়াজ আসছে, দেখ তো।
না, এবার ঠিক আছে।
ঠিক আছে,
হ্যাঁ হ্যাঁ,
ঠিক আছে। এখানে প্রিন্সিপালকে ফোন করে দিয়েছি, প্রিন্সিপাল বললো ঠিক আছে, মনে হয় ঘুমোচ্ছিলেন, বুঝেছিস।
আমি তো জানি এটা হবে, ওই জন্যই তো।
মনে হয় ঘুমোচ্ছিলেন, বললো ঠিক আছে।
Shreya Chakraborty, please stop presenting. Shreya Chakraborty, please stop presenting.
কি ক্যামেরার বাইরে থাকবি?
হ্যাঁ
হ্যাঁ
হ্যাঁ, আরে এত মুখে বলার কি আছে?
আচ্ছা, 4টে বেজে গেল তো, তাই বলছি।
আর 5টা বার, ঠিক আছে।
ঠিক আছে, 5 মিনিট দেখি, এখন তো সবাই ঢুকছে। শুরু করলে মুশকিল আছে। কোথায় লিখতে হয় গো?
Q&A বলে কোথায় অপশন দিতে হয়?
কোনো Q&A অপশন নেই।
এটা কি?
এটা টার্ন অন ক্যাপশন,
তারপরটা কি?
প্রেজেন্ট নাও,
তারপরটা কি?
তিনটে ডট ডট ডট।
কি আছে? ক্লিক করো।
চেঞ্জ লেআউট, ফুল স্ক্রিন, টার্ন অন ক্যাপশন, সেটিংস, রিপোর্ট প্রবলেম, রিপোর্ট অ্যাবিউজ, সবার নাম আর কলেজের নাম দিতে হবে।
এই যে প্রেজেন্ট নাও।
প্রেজেন্ট নাও কি করে
করো।
Pampaguho, please
না
turn
ভিডিওটা
off
অফ
your
করো না, তাহলে আমি
microphone.
দেখতে
কোথায়
পাবো।
ভিডিও এটা? Pampaguho, please turn off your microphone. Please, it's a request. হয়নি আমার। Pampaguho, please turn off your microphone.
এখানে তোমার ভিডিওটা দেখা যাচ্ছে তোমাকে?
আমাকে কি করে অনেক লোক চলতে যাও
কিচ্ছু অসুবিধা নেই।
খুবই সব। Pampaguho, please turn off your microphone. It's a request. তোমার সময় তো দেখে না। Pampaguho, please. আমার সময় এত বেশি।
এটা বলছি না, ভিডিওটা অফ করে দিলে ওইখান থেকে কি ভয় যায়? আমার ভিডিও অফ করেছে তো? আমার মিউট দেখাচ্ছে না, তোমার তো ভিডিও অফ আছে।
এই যে লাইট জ্বলছে তো।
আমার লাইট জ্বলছিল না। Send message in between.
ঠিক আছে।
ভিডিও কি অন করো না?
ঠিক আছে।
দেখছি না তো।
Pampaguho, please turn off your microphone. Pampaguho. Anyone who has the number of Pampaguho, please call and tell her. Pampaguho, please turn off your microphone. Live TV is it's a request. Oh, Pampaguho, please turn off your microphone. It's a request. Pampaguho, please turn off your microphone. Is there anyone who have the contact number of Pampaguho? Please tell her to turn off her microphone. Hello, Pampaguho, please turn off your microphone. Hello, Pampaguho, please turn off your microphone.
Amrita, will you start now?
Yeah, I'll start now. But Pampaguho, she has kept her microphone on and it's really loud. I'm just requesting her. I'm trying to find her mobile number. If I could call her and tell to,
Madam, if you are the host, you can turn off the microphone of Pampaguho.
No, I cannot. I'm unable to do it.
You can mute her.
Pompago is not looking here. Where is Pompago?
Okay, it's done, I guess, but someone else is still on with the microphone. Just give me a second. Okay. Can we wait for another just 2 minutes? People are still joining. I'm just requesting to wait for just 2 minutes. People are still joining. So in 2 minutes we could start.
Churala? Churala?
কারোর একটা ফ্যানের প্রচুর আওয়াজ হচ্ছে, প্লিজ।
হ্যাঁ চুরালা 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 চুরালা
আমি মিউট করে দিচ্ছি ওকে। Shunadi, shall we start? It's 4:07.
It's already 4:07.
Yeah.
Start.
Okay. Good afternoon, everyone. On behalf of Bashanti Devi College Chemistry Department, we welcome you all in this webinar on pharmaceutical chemistry. I am Dr. Amrita Mondol. I will be coordinating and moderating this webinar. As already informed, this webinar is divided into 2 sessions. Each session will be around 45 minutes, followed by 15 minutes discussion. Please send your queries in the comment box of the Google Meet. After the session, I will ask the queries on your behalf. I request you to please mute the microphone and video during the running of the session so that the session can run smoothly. And now I request our principal, Dr. Indrila Guho, to open this webinar. Madam, Indrila, Madam, Didi, আপনি আছেন? Hello? Hello?
Hello, Principal ma'am.
Principal Indrila ma'am, she is in mute and she is not responding at this moment. Sumanadi?
Hello.
Hello.
Shall we start then?
Hello.
Yes, please start.
Please begin first. Speak session. শুনতে পাচ্ছি না সাউন্ড।
হ্যালো, সঞ্চিতা কোথায়? অমৃতা, অমৃতা, সঞ্চিতা কোথায়?
ঠিক আছে।
ম্যাম তো এসে গেছেন। হ্যাঁ, ওকে ওকে।
Principal, I request you to please open the webinar and we could start. This is already 4:10. I request who have still not muted their microphone, please mute your microphone. It's very loud. It will be tough for us to go on with the webinar. Please, I request you all, if anyone. Hello? Hello, Shunchita, please start.
Please, principal ma'am is already in our group, so please request her to start the program.
Please call her and request her. She is in a mute.
She's in our group.
Yeah, I could see, but
Hello, ma'am. Ma'am. হ্যালো, অমৃতা,
হ্যাঁ,
প্রিন্সিপাল ম্যাম আমাকে কল করেছিলেন যে আমি রয়েছি, তোমরা আমাকে করো। এক্ষুনি কল করলেন।
আমি তো ওনাকে
প্রিন্সিপাল ম্যাডামকে আনমিউট করুন, কথা বলুন কি করে? মিউট আছে।
আমি ওনাকে মিউট করিনি অ্যাকচুয়ালি।
মিউট করা আছে।
উনি নিজে
তাহলে তুমি করো, প্রিন্সিপাল ম্যাম ফোন করলেন এক্ষুনি।
ম্যাডাম নিজে আনমিউট করে নিতে পারবে, ম্যাম যদি বলেন।
হ্যাঁ, বুঝতে পারছি না, ফোন করলেন তো, এখন আমি তো মধ্যখান থেকে ঢুকতে পারি না।
আমি ওনাকে মিউট করে রাখিনি. She has muted herself. So I can
only she can unmute herself. Nobody can.
Nobody else can unmute her.
Yes.
She is
So
unable.
Call her to unmute.
I cannot unmute her. If I have not muted her, I cannot do it. So maybe we should go ahead. We just call her and just, we should go ahead. Because it's already getting...
start.
Yeah, you should call her and tell, and then we'll start. It's better.
So we start. Then let's do.
Yeah.
হ্যাঁ বলো। আরে আমি তো ওনাকে ফোনে পাচ্ছি না, ফোন বিজি আসছে। 2 মিনিট দাঁড়িয়ে যাও, একবার দেখে নি, যদি হয়ে যায় ভালো, না দেখো করবো। আমি ফোন করছি, আমি ওনাকে ফোন করছি, যে উনকে হোয়াটসঅ্যাপে করলাম, দেখছেন না দেখি 2 মিনিট দেখে নিই, যদি পারেন ভালো, না হলে
Amrita, will you please start the session?
I have asked Sanchita
Technical
to,
session.
I asked Sanchita to please introduce our speaker so that we could start our session.
I think Indra Guru is present.
হ্যালো ম্যাম, আপনার না মিউট আসছে অ্যাকচুয়ালি ওই ইয়েতে।
She has left the meeting.
আপনি ওই ইয়েটা আছে, আপনার
I think
বাটন আছে,
Sanchita, please
একটা ওই
introduce our
মাইক্রোফোনের বাটন
speaker
না রেড হয়ে থাকে, এটা কাটা
and
দেওয়া
start
আছে।
the meeting. Sanchita, please. Sanchita,
আপনি মোবাইল থেকে
will
করছেন
you
তো?
please introduce our speaker and start the meeting?
তত হয়ে যাওয়া উচিত।
Okay. Good afternoon to all. Now we are going to start our main
ওটা
session.
দিয়েই হবে।
Today we have 2 lectures. First lecture is on drug design and discovery. The lecture will be delivered by
হ্যাঁ, সেটাই,
Professor
ওখানে
C.M.
just এখন
Hossain.
মিউট দেখাচ্ছে। অমৃতা দি তো
Professor
বলছে যে
Hossain did his
unmute করা
B.Pharm and
যাচ্ছে না।
M.Pharm degree from Department of Pharmaceutical Technology, Jadavpur University. He got gold medal in
আচ্ছা,
the M.Pharm program.
আপনি কি
He
তাহলে
did his
ফোনে বলে
PhD
দেবেন?
degree
আমি
in
ঠিকানা আমার বলতে
pharmacology from the Department of Pharmaceutical
মানে আমি
Technology,
আমার
Jadavpur
মাইক্রোফোন থেকে
University.
করে দিচ্ছি, আপনি এখানে
He
বলে
is
দিবেন।
former
মানে আপনি ফোনে
postdoctoral
যদি
fellow
কথা
in
বলেন
School
সেটা
of
ওখানে
Medicine,
শোনা যায়।
University of Florida, USA.
আচ্ছা, আমি
Currently, he is
আপনাকে
working as professor and head of the department in Department of Pharmaceutical Science and Technology. He is actively engaged in research work, and his research interest is on drug discovery, on infectious diseases, diabetes, and liver cancer. He has published different research papers in peer-reviewed international
ম্যাডাম
journals
বলছেন
like
একটুখানি অ্যানাউন্স
Nature
করো,
Group,
আমি ওনার
Elsevier,
কিছুটা গন্ডগোল আছে, আমি আমার
Springer,
মাইক্রোফোন দিয়ে
etc.
উনি ফোনে কথা বলছেন।
He is editor of a couple of international journals and contributed as a co-author of several books
একবার বলো
and
এরকম,
book
কারণ ম্যাডাম
chapter
তো
on
থেকে
relevant
এসছেন বলে
topics.
ম্যাডাম।
He is a member of Indian Pharmaceutical Association. So I request
ম্যাডাম
Professor C.M. Hossain to start his lecture.
দেখো দেখাচ্ছে ওখানে।
Sanchita, can you hear me now?
Yes,
Ma'am,
yes, you
of
are
course.
already present here.
Okay. Ma'am, please come and inaugurate the session. Yes, ma'am. Hello?
Hello,
Can you
ma'am.
hear me?
Yeah.
We can hear you, but we cannot see you.
Can you see
I think
me
the
now?
video is off.
Can you see me now?
No, ma'am, no.
No.
Can you see me now?
No, ma'am, we are not seeing you.
Able to see
ম্যাম
you.
দেখতে পাচ্ছি আপনাকে,
হ্যাঁ
দেখা যাচ্ছে। এখানে হচ্ছে যে মানে যিনি কথা বলবেন তারটাই সামনে চলে আসবে, আপনি কথা বলুন, অটোমেটিকলি ভিডিওটা চলে আসবে।
আচ্ছা দেখো এবার দেখা যাচ্ছে
No, ma'am.
আমার স্ক্রিনে আসছে সবার মোটামুটি
Yes, we can see you. Yes.
আসছে, একটু কথা বলুন, চলে আসবে।
এবার দেখো shall I start?
Yes, yes.
Okay, ma'am. May start.
Yes, ma'am, please start.
On pharmaceutical chemistry. This is a pharmacology section of semester 4. I welcome Professor Dr. M. Hossain, who will speak on drug design and discovery. I also welcome Dr. Niladri Shekhar Roy, who will speak on fermentation chemistry. It is really a privilege to work with Board of Studies of Calcutta University because we are collaborating with Board of Studies Calcutta University. We had a plan to hold it not virtually but in our premises, in our auditorium. But all of you know that due to lockdown, we could not do it. Since 15th of March, we are closed it. so we had to go for the webinar. And for faculty members of Pharmaceutical College and also of other institute colleges, we immensely benefited from this webinar. Full to the chemistry department of and also the head of the college, and Dr. Aditi Shankar, who is the coordinator. She has also helped to organize this webinar. I wish good luck to this webinar, and I also wish I'm thankful to both of our presenters, Dr. Hussain and Dr. Roy. And I think we should start without further delay. Thank you very much for giving me this opportunity to inaugurate the webinar today. Thank you very much.
Thank you. Well, let's go directly to the technical talk.
I have already introduced Professor Hoshen, so I request Professor Hoshen to start his lecture.
Okay. So let me share my PPT first. Can you see my PPT presentation?
Ma'am, you can now turn off your video.
Can you see, ma'am? Can you see my presentation?
No,
We cannot see.
Please click on Dr. Hoshen's ID, then you can see his PPT.
Dr. Hoshen.
Yeah, Dr. Hoshen. There's a name on the left-hand side, but the people are there. They have
Dr.
added
Hoshen talks.
Yeah. Then you
Can you
can
see my PPT?
No, I can see you, but
Sir,
I think some other people are in the presenting mode, maybe.
sir, have you clicked the button on present now?
Just I have clicked present now.
Okay.
Then enter screen.
Enter screen. Yes.
Then already I shared. After that, after what should I do after that?
No, it should share the screen, sir.
Okay. Then.
But your video is only coming. The screen is not being shared.
yes, we can only see you.
Is it okay now?
No, not visible.
No, sir.
So are you doing it from your laptop?
Yes.
Okay, then in your laptop you'll be finding share here. Then you select the second option, new window, and probably your presentation is on your desktop.
Yes.
So it directly go to your desktop. Then you click on this window. So from desktop, you click on your PPT and then you click on the share button. It will come live in the screen.
Okay, so my PPT is already open.
It's open. Then the share will come in the below of the screen.
Okay, again I'm doing now. Okay.
Yeah, because same thing I did yesterday in the evening in Hangout Meet.
Or better, I'm sending you the PPT. You can do this. You enter screen, then share. Already I have shared. There is no button for share. Already I shared. Is there anybody in the presenting mode?
Sir, please share the screen. You just did it a few hours back, so please share the
Yes,
screen.
you did it.
Yeah, we just
Share
checked
the
it out. Please share the screen. I asked Mr. Shumit Pal and Mrs. Rupa Roy to please turn off their video. Your videos are on. Dr. Shumit Pal and Mrs. Rupa Roy, please turn off your video.
Amita ma'am, should I send you the slide? You'll share it?
Please send it to Shumik.
What is the email ID, please?
Reach Shomik, RESCH
Just a moment.
Yeah, sure.
Just a moment.
Dr. Shubhanna Chakraborty, please turn off your video. Madam, please turn off your video. You are
What is your email ID?
Yeah, it is Rechromic. R-E-A-C-H.
R-C-S.
R-E-A-C-H. Reach.
R-E-S-C-H.
S-A-M-I-K.
S-A-M-I-N-K.
S-A-M-I-K. Rechromic@gmail.com.
Reach, R-E-A-C-H.
R-E-A-C-H, yes.
R-E-A-C-H. Samik. S-A-M-I-K.
M-I-K.
At
gmail.com.
Okay, sir. I've just sent.
Okay, sir. Just take me.
Okay.
Amrita, can you hear me?
Yes.
Amrita, Dr. Kinjal Kini Vishwas, Principal, Muralidhar Girls College, wants to join.
Please.
I am sending the link.
I am sending the link.
No, but her request is denied. Please look into the matter. She's trying to join.
Can you see my slide
Yes,
now?
yes. We could see.
Yes, sir. It's visible.
It's working.
It's
Okay,
visible.
well, let me start. Good afternoon, everybody. First of all, I would like to convey my thanks and regards to Dr. Indrina Guho, Principal of Pharmaceutical College, for inviting me and giving me the opportunity here to deliver my talk. And I would like to thank Dr. Sucharita Rai, a faculty of Chemistry, Department of Pharmaceutical College. And also I would like to thank the convener, Dr. Shumana Chatterjee, and Dr. Amrita Mandal, who is coordinating and hosting this particular webinar organized by Baswanti Devi College on Pharmaceutical Chemistry for undergraduate course in collaboration with US UG Study Calcutta University. So in syllabus I have gone through that there is two portions. One is on drugs and pharmaceuticals, particularly drug design, drug discovery, and drug development. And after that, lots of synthesis are there. Synthesis of compound, particularly like cardiovascular drugs, anti-anginal, antiviral. A lot of drugs are there. Just synthesis are there, and second part is fermentation part. So I would like to discuss on the first part. So as we are all people are from the chemistry background, without chemistry people, the drug discovery is not possible, because the first step to synthesize or isolate or to characterize the particular compound, we need pharmaceutical chemist or scientist for this work. So here I will just show you that. Am I audible to all?
Yes.
Here I will show you that they're the first do no harm. It is good remedy sometimes to use nothing. That is said by Hippocrates in BC 463-55. That's do not use. It is good remedy sometimes to use nothing. Another quotes, that is what is medicine, as by Charok Samhita. The medicine is the right one and pure one which cures a disease, physical, mental, and spiritual, and doesn't give any adverse drug reactions and doesn't create any other disease. Very important, because nowadays what medicine we take, a lot of medicine that cures some disease, parallelly it causes another disease. That is actually not required. Another quotes from Paracelsus, he's a great scientist. He said everything is poison. That depends on the dose you are taking. So dose alone decides that something is not poison. Even water is also poison if you take in excess amount. So let me start. What is drug? A pharmaceutical drug or medication or medicine, what you say, this is a chemical substance basically. Basically, this is a chemical substance. We convert it as a drugs by several process or development process. So this chemical substance to treat, cure, prevent or diagnosis or disease or to promote well-being, that is called drug. And what is drug design? Basically, the chemistry people, they do the drug design. After that, a lot of processes are happened in drug discovery in pharmaceutical industry. But initially, drug design is important. So what is drug design? Drug design often refer to the finding new medications based on the knowledge of biological target. Actually, we have to see what is the target, what is the disease, what is the abnormality going on in our body. So important thing is we have to identify the target, then we have to design the drug accordingly. The drug is most commonly an organic small molecule, mainly, that activates or inhibits the function of biomolecule, such a protein in the body, which results in therapeutic benefit to the patient. Actually, drug, when you administer on body, it binds to the protein or enzyme on a receptor. That is a target inside the body, and it produces some therapeutic effect and recover the patient. In the most basic sense, drug design involves designing of the molecules that are complementary in the shape and charge. So design is design of the drug based on the shape and charge of the biomedical target. The target in our body that should have similar structure in shape or charge with the biomolecules which they interact effect. And you also sometimes you have to deal with this term during the drug discovery, because a lot of chemistry people, they work in the pharmaceutical industry. So this is drug delivery. So after designing, after discovery, drug is delivered in a form in the body. So this refers to transporting a pharmaceutical compound in the body in desired concentration at target tissue to produce therapeutic effect. Some of the common routes of the administration, like enteral, like when we take some oral capsule and tablet, whether we are giving some injection, inhalation, transdermal patch, nanoparticle, liposomal drug delivery. So in which form with the drug is delivered in the body, that is dealt by the drug delivery section. So drug development is the process of bringing a new pharmaceutical compound to the market once a lead compound has been identified through the process of drug discovery. It includes preclinical research, clinical trials on humans, and regulatory approval with a new drug application to the market. So there are three terms you see. One is drug design, another is drug discovery, and later one is drug development. So drug design first, then after drug design, drug discovery. After discovery, there is process that is starting the animal. Then after getting successful data from the animal, we go for the clinical trial for human to administer the drug to the patient. So that is clinical trial. So the preclinical in animal testing and human testing, this part is called the drug development. So drug design, drug discovery, and drug development. In the past, earlier days, drug has been discovered by identifying the active compound from traditional remedy. Usually, in earlier days, most of the people use some herbal drugs. They isolate some drugs, or they used to use some whole plants for remedies, or by serendipitous discovery, like by chance. A lot of drugs or a lot of discovery happens by chance. But now we know the disease, how the disease is happening, what is the pathology, what is the molecular reason behind any disease. We can understand at physiological level and molecular level. Also, shape of a molecule at atomic level is well understood. Information of human genome also well understood. Hello, is it audible? Hello. Hello?
Yes, sir.
It's completely audible.
It's audible. Please continue, sir. Please continue.
Please continue.
Okay,
It's
okay.
going very fine.
Okay, thank you. So now I'm just giving a brief history of the drug discovery. You see, it will be more interesting because everyone likes history, but the background of the drugs. You see, in 1919, the pre-1919 actually, because most of the drugs came after 1919. People used to use some herbal drugs or some by chance discovery. In 1920, we got some vaccine, some vitamin. We say 1940, the antibiotic era, because most of the antibiotic like penicillin is discovered by Alexander Fleming in 1940, and a lot of R&D work boosted up due to the World War II. After that, 1940, 1950, there's some new technology like discovery of DNA happened. After that, in 1960, some breakthrough in the etiology, like where we try to know what is the reason behind it, what is the pathology, what is the molecular level. We try to understand in 1960. In 1970, there is a rise of biotechnology, use of IT, and 1980 there is commerce. There was like commercialization of drug discovery, combinatorial chemistry, and after that in 1990 robotics, automation, and nowadays we are in the era of some artificial intelligence, which helps a lot for the drug discovery. You see, during the drug discovery of drug design in the industry, pharmaceutical company, they actually start with the many compounds. Like when they synthesize or design to discover new drugs for a particular disease, they synthesize like libraries. If you see, initially they synthesize 10,000 compounds during the design, but when it comes to the preclinical level, means for the testing of the animal, it reduces to 250 compounds. So you see, lots of compounds are wasted. And after a successful result from the preclinical studies, 5 compounds came for the clinical trial for the human testing. And from 5 compounds, 1 drugs may be selected or may not be selected. You see, there's a lots of wastage of money and time both if there is a failure of the drug discovery. And you see here, from drug design to preclinical study, it takes almost 6.5 years, like 7 years it takes from synthesis to preclinical, like to animal study. And human study takes almost like 5 years, and from clinical trials, from approval of the drug in the market, it takes 1.5 years. So total 16 years is taken for the discovery of single drug. Can you imagine? And if this process fail, so there is total wastage of money. It's a huge money. That's why the cost of cost of the medicine is very high. Because cost of the medicine is very high, because most of the multinational companies, they try to discover new drugs, and most of the local company in India, they use some generic drugs. So those people who actually involved in the R&D for discovering new drugs, they have some patents. So they also invest lots of money, and they face lots of failures in drug discovery. So they try to recover the money through the drugs in the market. So that's why the cost of the medicine is very high. Now, what is the techniques of the drug discovery? Older approach was, what we people do, they tried to isolate some natural compound like morphine, like atropine. These are the like quinine. All of these drugs are used for the treatment of several diseases, but those are isolated from the herbal product. And some people use some traditional medicine like whole plant or whole leaves or extract of leaves, and some barks are used by some tribal people for the remedy. And older people, and some modification of structure of known drugs, like there's a drug ciprofloxacin, we try to modify the structure, or repurposing of the drug. That is actually we are trying to practice currently in COVID-19, because we all people know about the hydroxychloroquine drugs that is used for the rheumatoid arthritis, autoimmune drug. And this drug actually currently tried to be established to treat the COVID-19, because this is repurposing. Like this drug already used for the treatment of another disease, they're trying to establish it for a particular another disease. So this is the repurposing of the drug. And another techniques, this is the newer techniques. Currently, most of the research lab in the United States or Europe or in India, definitely in modern lab, they try to develop many drugs by molecular modeling, computer-aided drug designing, some biotechnology, recombinant DNA technology, which is used for the production of insulin for treatment of diabetes, and some combinatorial chemistry. The combinatorial chemistry, that objective of this combinatorial chemistry method is to generate huge amount of compounds very quickly in a single process. So for the drug discovery, very important thing is the therapeutic target. We have to understand what disease, like we have to discover the drugs against particular disease. So we have to select the disease. If you want to select the disease, we have to select the target. Target means the protein, that enzyme, the receptor, that abnormality happens in our body. So once therapeutic target is established, we try to discover the lead discovery, lead optimization by using a combinatorial chemistry here. And after that, development stage happens and drug comes in the market. So you see here, the approaches to the drug discovery, like we have to, you see this is what we see, that this is the target. So this man is targeting these fruits. So we have to targeting like this, that this, but for this particular disease, we have to discover this drug. So molecular target is very important. After that, the selection of the lead compound and validation of lead compound, I'm discussing it here. So let me discuss you that the target, what kind of target in our body, like we have lots of enzyme, lots of receptor, nucleic acid, ion channel, common targets in our body. Most of the drugs either binds to the enzyme, nucleic acid, ion channel, transporter. In this way, they produce action in our body. So I am talking of why we are talking about this, because chemistry people should know some targets. Pharmacology people should know some synthesis procedures. So this is an interdisciplinary work during the drug discovery. So all people should know this. The target selection in drug discovery is defined as the decision to focus finding an agent with particular biological action that is anticipated to have therapeutic utility is influenced by complex balance of scientific, medical, and strategic considerations. Target identification to identify molecular targets. So what is first target selection, then target identification, then target validation? That means target selection means which targets I have to hit. Like G protein coupled receptor, a lot of receptors are there. G means glutamate. Abnormality of those enzymes may cause many diseases in our body. Many hormones and factors, ion channel, nuclear receptor. Most of the steroid drugs that binds with the nuclear receptor intercellularly. That's why long-term administration of taking of steroid causes lots of side effects. This is present inside the cell, this kind of receptor. So this is a very harmful, this receptor is present inside the cell. Most of the receptor present on the cell surface, but this receptor only present inside the cell, and steroid binds with it and produces lots of side effects in our body. After target selection, target identification, so drugs usually act on either cellular target or genetic chemical in the body, which is called target, which are believed to be associated with disease. So what we do, we use some techniques to identify and isolate individual target to learn more about their function and how they influence disease. Compounds are then identified that have various interaction with the drug targets that might be helpful in treatment of specific disease. So these are the target. The targets may be in the cell or in the maybe DNA. So in this involves identification of target receptor or enzymes, whereas some people approaches the focus is at the gene or transcription level. So any drug, when we take like in tablet or capsule or any formulation, this drugs either it binds with the cell or it binds with some gene like DNA. So you also heard that anti-cancer drug, most of the anti-cancer drug, they bind with the DNA actually. Drugs usually act on the targets which are associated with the disease. Some drugs, they directly bind to the gene. So genomic study is very important. So many diseases that may be developed due to the genetic predisposition or genetic, so we must study the genes. So sequencing of human find out new targets. So sequencing, we have to know the genes of human and also pathogenic organism like the virus COVID-19, that's coronavirus. We should know the genome. We have to know that different strain, different species. We have to know the details of the genome. Then we can discover the drug. And we also have to know the details of the genome of different race, different species in respect of geographical location in the world. Based on the 5 or 10 linked protein per gene, proposes that number of potential drug target may lie between 5,000 to 10,000. You see, number of potential drug targets in our body, it ranges from 5,000 to 10,000 in our body. So any target, there is any abnormality occurs in any target that produces disease. Now come to the point, proteomics. So chemistry people definitely heard this name, proteomics. This is very interesting topics nowadays. because more 91% drugs bind to the protein in our body, because we are made of protein. Because protein comes from gene. Gene is translated from to the protein, and we are made of protein, and protein is regulating a lot of function in our body. Therefore, analysis of proteins like protein-protein interaction, protein nucleic acid, protein ligand interaction will be utmost importance to target drug discovery. Target identification with proteomics is performed by comparing the protein expression level in the normal and disease tissue. After that, we have to know the bioinformatics. This is a very important domain in the research of the drug discovery, because if you know all the genomic sequence, it will be very easy to understand that which chemical can bind the particular target. So for the target identification, we have to know the gene, computer screening of chemical compounds, pharmacogenomics, because drug may affect or influence lot of side effect or effect on the basis of the gene. Maybe some drugs may work very well, like paracetamol may reduce temperature very quickly in me, but if 500 milligram paracetamol is given to you, it is not working. So maybe ciprofloxacin is working on me and it is not working on you in particular dose. So a lot of genomics factors are there that influence the action of the drug in our body, and that's to be studied. And we can compare the entire genome of pathogenic and non-pathogenic strain of a microbe and identify gene and protein associated with the pathogenesis of the disease. And target validation, to select the most likely to be useful, like we have to select, we have to identify, but we have to validate it. Otherwise, we cannot use it. So for validation, to select most likely to be useful is the development of new treatment of the disease. Researcher analyze and compare each drug target to other based on their association with a specific disease and ability to regulate biological and chemical compounds in the body. So what we have to do, we have to understand the targets, and we have to understand the association of the targets with the disease. Tests are conducted to confirm that interaction with the drug targets are associated with desired change in the behavior of the disease cell. The scientist can then identify compounds that have an effect on the drug selected. So this is what I talked about. This is the target selection. Now about the lead identification, that is the actual function of the chemistry people, because I was engaged in the drug discovery in the US in the lab, because we are working in the drug discovery against infectious disease particularly, and it is in the clinical trial right now, and we got patent in the US. So what we did, we helped lot. We got help lot from the chemistry people for synthesis of the particular compound. So here you see the lead compound means the chemical compound or substance is on that is believed to have potential to treat disease. Laboratory scientists can compare known substances with new compounds to determine the likelihood of the success. So what we do, when we discover, we try to compare with the existing one. The lead compounds sometimes develop as a collection or libraries or individual molecules that possess properties needed in a new drug. Testing is done in each of these molecules to confirm its effect on the drug. So how do we identify the lead compound? Maybe we get from the bacteria, like lots of antibiotic we got from bacteria, molds, or plant extract by nature. Maybe de novo synthesis. We can synthesize the new molecule in the lab by laboratory synthesis or computer modeling, or high throughput screening. The test of thousands of chemical compound against target to identify any might be promising for the treatment of particular disease. For lead discovery, identification of small molecule modulators of protein function and process of transforming these into high content lead series. So in lead discovery, what we do, either we separate from the mixture, we separate or isolate particular compound of the mixture, or we separate a compound from the impurities. Many compounds may be obtained from the impurities. So a lot of scientists now are very much focused on the impurities. That many compounds may present in the impurities. They try to isolate. And in vitro chemical synthesis and some biosynthetic intermediate. Now regarding the combinatorial chemistry, because this part helps us for the rapid synthesis for computer simulation for large number of different parts of structure related molecules. I am not a chemistry people, so I do not know a lot, like I'm not expert on this simulation, but I have some superficial knowledge. Such new leads optimization of target affinity and selectivity. And ADME part, what we do as a pharmacologist, we do this part: absorption, distribution, metabolism, excretion part of the particular compound, and how the toxicity of the drug can be reduced or eliminating the side effects in the body. And also drug can be developed by the assay development by measuring the activity of the drug. So all you know about this high throughput screening, because I think all are learned faculty members here. So I think all people know about this high throughput screening, but this is currently replaced by in silico mechanism of computer aided drug design or docking. All this current facility or current developed system has replaced this particular automation system. So in high throughput screening, what happens? Screening of drug target against selection of lot of chemicals and identification highly target specific compound. So this is the computer aided drug design, which will be very much helpful for the students of pharmaceutical chemistry, undergrad or postgraduate research. This is very much used for the computational approach for developing a new drugs, to discover, develop, or analyze the existing drugs, or modify the structure of particular drug, similarly biological active molecules. This is actually the ligand-based design. Ligand are studied, ligand-based computer-aided drug discovery using software. Approach involve the analysis of the ligands known to interact with the target of interest. So in this particular design is like QSR, like quantity structure activity relationship with strategy of this essential importance for chemistry and pharmacy, based on the idea that when we change the structure of the molecule, then also the activity or property of the substance will be modified. As you know, the bench diazepine group, that is for the alprazolam or Valium, the sedative drug, which is used to produce sleep. We take some anti-anxiety drug. So that in that structure, benzodiazepine ring is present, and their functional group chlorine is there, that is responsible for inducing sleep. So if we modify the chlorine group and by other groups, its activity will change. Another recent system for computer-aided drug designing is docking. This is a receptor ligand interaction study. Actually, that ligand binds to the receptor, and there's this crystallography structure of the receptor is there, and that is interaction is studied actually. Now, after lead discovery, we have to optimize, like we have to validate whether lead is working or not. So lead optimization compares properties of various lead compounds. So if we optimize like 100 compounds, we have to compare which one is good. So we have to provide information to the pharmaceutical company to select this particular is good, company is good, this particular is compared to very good as compared to that. So we have to establish this, the chemistry people, that this compound are with greatest potential to be developed into safe and effective medicine. Because our objective to produce not only the medicine, our objective that is a medicine which safe and effective without side effect. So often during this same stage of development, some lead optimization done by studying on some organism in vivo or in cell in vitro test tube to compare various lead compounds and how they're metabolized and affect in the body. This optimization, what happens in optimization, the structure are altered. Like there is a structure, we optimize it by altering the structure of the lead compound to improve the property. If we change the structure, the property may be improved. If we change the structure, the interaction of the chemical in our body to produce side effect, that will be less. That will be helpful reducing the side effect maybe. So a structure may be altered to improve the efficacy or reduce the side effect in our body. Or we can produce some analogs of the initial lead compound and test it by biologists to the effect of the analog on the biological system. chemist people, the chemist scientists, they take this information to the biologist. So what happens? So I have that direct experience because I worked on some chemical compounds synthesized by particular pharmaceutical industry, and the chemist people, they send us the compound, and I study on the animal against some infectious disease. So when I didn't get any good results, so I talked with some chemists. There's a meeting, and I tell that this kind of side effect maybe there are a lot of side effects, like it causing some angina or causing some blood clot in the heart. So then chemist people, they think that what to do. They modify the structure. They modify the structure, and they again send the modified structure, and then we screen the molecule against the disease. In such a way, the drug discovery process goes on. So in process, the lead compounds go through a series of tests to provide an early assessment of the safety. So most important thing is that we have to establish the drug is safe or not. First point is whether the drug is safe or not. If it is not safe, we'll reject it. So what we do, we establish for the safety profile of the drug, and we see whether it will be absorbed in our body, whether it is distributed properly in our body, whether it is metabolized and excreted in the body. Otherwise, it will accumulate in the body, and toxicological property will develop. So what is the successful drug? After drug design, the successful drug must be absorbed in the bloodstream. It should be absorbed very easily in the bloodstream. It should be distributed properly, uniformly to the site of action in the body, where actually disease has progressed. That should be metabolized efficiently and effectively, and should be successfully excreted from the body, should not demonstrate toxicity. It will help the optimization process, help researchers prioritize lead compound early in the discovery process. So in this way, what we do, prioritize, we optimize, we validate the compound which is better. This is a continuous process. So the ADME, the adsorption, distribution, metabolism, excretion, toxicology study are performed in the living cell, in animal, or via computational model. All you know that the QSR, quantitative structure activity relationship, or SAR, that is done for the drug design, that helps us to identify the pharmacophore. Pharmacophore, that is a group that has actually particular biological activity against particular target. The pharmacophore is the precise section of the molecule that is responsible for biological activity. That enables to prepare more active compound and allow us for the elimination of excess functionality. So through QSR, that help us to select the particular very good compound, good, safe and efficacious compound, and also help to eliminate unnecessary compound which have excess function, which is not actually required by us. Now come to the in silico screening. So we know the computer simulated screening of chemical, all are in silico screening, that helps in finding structure that are most likely to bind to target and filter enormous chemical space, and it is definitely economic than HTS. So this part, before this, what I talked, this is completely drug designing. After drugs designing, we have to apply to the some regulatory body for INDA, like investigational new drug application, that we want to investigate. We have a drug, we have a very good drug, and we saw that the drugs may bind to the particular target and reduce disease, and we are trying to establish whether it has a good pharmacological action. So application should be sent to the DCGI, Drug Control General in India. They will approve the INDA and will start the preclinical study for the clinical trial. You see, this is the animal study we do. In animal study, what we do in biochemical assay, basically what we do in animal study, we develop some animal model. Like if we discover diabetic drug, we develop diabetes disease in animal. If we develop cancer, so we have to discover some cancer model in the mice. If you want to discover the drug against COVID-19, we have to inject this particular virus through the tail vein of the mice and develop corona, like COVID-19 infection in the mice, and we have to test it. Otherwise, we will not get a particular drug against it. So in this way, we develop some animal model. Before animal model, we do in vitro study like biochemical assay, some cancer cell line, a mucosal model. This is the preclinical study. This is the study in animal. What we do, we develop many disease model. After that, or not. Parallelly, we also see whether drug has. We see whether drug has efficacy, whether drug is acting against disease or not. But in toxicology, we see whether drug has potential side effect, whether lifelong the take therapeutic effect means if you all this study are performed in particular phase 1 study in the hospital. In phase 2, the people taken number of patients are 50 to 300. This is very important phase. Here, patients are taken. Like if you want to discover the anti-diabetic, we have to arrange 300 diabetes patient and controlled single blind technique. And what we do, we test safety of the drug here, we test efficacy of the drug, and we also see drug toxicity and also drug interaction, like which drug, like